So you may have a strand of DNA, which is, let's say, for your hemoglobin, which I'll abbreviate HGB. HGB, hemoglobin. And there are 4 different parts to hemoglobin, 2 pairs of things, hemoglobin A. And it turns out that at some time in our history, it got duplicated. And then, because this was functional, this one could change and mutate, and it did. So we now have sections, we now make hemoglobin as tetramers around the middle of an iron, and it's the iron that binds the oxygen. And these are A, 2 A's, and these are B. No, that's not very good. A, B, B. And so we've duplicated our hemoglobin. The same thing is true for our clotting cascade. People who argue that evolution doesn't work look for complex things that would be difficult to explain through evolutionary processes. And one of the ones that they talk about is the clotting mechanism, because it involves 12 proteins. In order to make the little clot that filled this space on my arm after I went blam, 12 different proteins had to work in concert, and actually more, but at least 12 to form this little clot. And the way that they were formed, if you look at the DNA, or if you look at the protein structures, you'll find that there actually are a lot of similarities between 1 and 2 and 3 and 4. It looks as if there was a small subset originally, which got duplicated and duplicated and duplicated to end up with these 12, which have very slightly different functions but work in concert. And natural selection loves duplication of genes, because what that means is you can have an A gene which is doing its thing, properly coding, and you can have the B gene which is free to change its function, free to be completely deteriorated into nothing, or free to be excised and thrown away. I'm seeing some blank stares, so I think I better stop.
Yeah. While you're talking about function, separately from what?
Function comes from the structure, from the 3-dimensional structure. But it's extremely complicated. And we're, when I was getting my master's degree, there is a brilliant woman who was using all the computers in the computer lab in the off hours, because she was looking at how proteins fold and trying to determine what was the best fold given a protein structure. We still don't have that kind of knowledge, and we really need it. We are at the end of our experiment. And what you do is you reach inside here, and the stuff that's in between, or that is willing to come up into the alcohol layer, is the DNA. And I'll leave that for people to come and take a look. You can see last year's DNA, quite a bit. That was about 1/4 of a banana's worth. But I think I'll stop there. Questions? Comments?
I don't know. I'm trying to find DNA in that. Is that
the fuzzy little white stuff at the top?
It settles out, or
it comes up.
Oh,
yeah. All the stuff that is now on the top of the glass.
Oh,
this is DNA right here.
Oh yeah. So if we looked in the microscope, it would.
Well, a microscope wouldn't be enough because this has been DNA that's been, treated roughly. So you're not going to see chromosomes and that kind of thing. But if you put it through a DNA nucleotide sequencer, you could do that.
Now everybody's DNA is different, right?
Mhm.
It's like fingerprints. But my son's DNA probably very close to my DNA, right?
He has half of your DNA.
Oh, that's the way they do it.
Right,
That's
right.
what they did with the Obama and then the aunt of Obama, you know.
Yeah. Well.
Yeah, I mean, they check it out, right?
Yeah. Yeah. You know,
More
let this in.